Okay, good morning, everyone. Happy Friday, this first week of classes. I hope you've all survived your first week. I think I'm barely surviving, but I still have the rest of the day to kind of get through. So I'm still trying to figure out what's going on with the screen sharing glitching. I'm trying something else now with sharing the screen. Hopefully this will work a little better. But at any point during the lecture, if it looks like the screen sharing's messed up again, like I've moved on, or it sounds like I've moved on to the next slide and you guys still see the previous one, you just go ahead, unmute yourself, let me know, because I think if I stop the share and start it again, it seems like it fixes it. Okay, but let me know, because from my end, I don't see anything different. And to me, everything looks good, right? And so it's kind of a shame if I'm just rambling on and on about something that you guys aren't even seeing. Okay, let's talk a bit more about the way the assignments are set up in this class. Right? So there are two sections, section 1, section 2. Those of you in section 1 are doing the more traditional, non-writing intensive focus credit version of the class. Those of you who are in section 2 are doing the WI credit version of the class that involves a semester project. I will introduce the semester project to those of you who are in section 2 next week. Okay. You don't have to worry about it for now. For now, I want to talk about stuff that's common to both sections. So there are 3 types of assignments that are pretty much week to week. Okay, so weekly assignments. First off are the quizzes. So pretty much every week there's a 10-question multiple choice quiz that's set up in Lalima for you to log into, Lalima tests and quizzes, and you'll see it there. It's timed for 20 minutes. You just go ahead, and whenever you feel like you're ready to take it, you go ahead and you take it. And those quizzes are due Wednesday at 10:30 a.m. So in other words, the following Wednesday at class time, you need to have finished your quiz and gotten it turned in. And the quiz is meant to kind of go over stuff that we had been talking about the previous week. All right. Today, I will post both a practice quiz and the first real quiz. So I would recommend that you try the practice quiz first. It doesn't count. It's just to make sure that your Lalima is working fine. I think most of you have taken online quizzes in Lalima by now. But, just try it, make sure it works, and then make sure you take the first real quiz, which will be due next Wednesday at 10:30 a.m. Then every week we alternate between the 2 other types of assignments. Some weeks, like this week, we have a homework assignment due. So hopefully, right, you're already aware that starting at the beginning of the week, I made this first homework assignment due. And I think they're always just going to be like 4 short answer questions that you can do anytime during the week. But you need to make sure you submit them by Friday night, 11:55 p.m. Right? So on the weeks that alternate with that we don't have homeworks, we have an article summary assignment. One of the things that I really try to emphasize in this class is for you to get out there and look for examples of current published scientific reports that elaborate or give examples, real-world examples of the different types of topics that we are studying in the class. I think I mentioned to you before, the textbook's a few years old. I think it's like a 2014 textbook. For some types of subjects, not much changes in like 5 years, 6 years, but the field of cancer is rapidly evolving. So some of the things in the textbook that are just sort of presented as kind of like almost like someday we will know more about this or something like that. In reality, in the intervening years, right, there's been tremendous progress on that. So a great way for all of you to feel really kind of like you're on the cutting edge of understanding where the field is with these cancer topics is to go out and find examples of current papers. What do I mean by current paper? It doesn't have to be a 2020 paper, right? But it should be something that's not older than the textbook itself. So I think something like no older than 2014, let's say, okay? I will provide a set of documents on Laulima that help illustrate what I'm expecting from you from this article summary. But the way it works is you'll find a paper, you will read it, right, and you will write basically a one-page kind of summary that explains something about the background of the paper, something about how the study in the paper was set up, what the results were, and how you feel this ties in with what we were learning in the class. Okay. Again, those instructions, I will give you very explicit instructions in the form of a document that you can find online. You write those up, you turn them in by Wednesday night. Okay, so you have until Wednesday night to turn those in. The reason is on Fridays of those article summary days, so every other week, right, we do this article summary thing. Instead of a normal lecture, like what we've been doing so far, we will do an article share session where you get to talk about the paper that you chose and kind of share with the rest of the class, what it is, right? Your TAs, so Alexis and Zoe, right, they need some time to be able to look at all the topics that the class has chosen for that week to help facilitate what the discussion will be like. So that's why you guys have to turn in your paper by Wednesday night so that Alexis, Zoe, and myself can have Thursday to kind of look at all the topics and put together kind of a meaningful discussion to facilitate with you guys on Fridays. All right, so for now, keep that in mind. So that means starting from today, you should go out and start looking for a peer-reviewed paper, and they must be peer-reviewed papers. So do not go to Yahoo, right, and find some article that appeared on Yahoo that talks about things like, smoking and cancer or something like that, right? We want real peer-reviewed primary literature, right? Because that's something that I want to train you, since you are all upper division students, seniors finishing up your MCB or MBBE degrees. You want to have exposure to get used to reading and being able to communicate what you're finding when you do literature searches. All right, so look for that info on Laulima. I will make sure that info is up there after lecture. All right, so yeah, when you go to Laulima, it'll be up there something like this, right? There'll be a folder for the article summary assignment. There will be the instructions. There will be something that explains how they're graded. They will explain things like the rubric that are used to sort of score your assignment. There are also some suggestions for topics for this first couple weeks in case you're at a loss as to what to do. I've given you some suggestions here that you might want to try. And I've given you some samples, right? Samples of past students' article summaries so you can see what we're looking for and how those students' works were graded, right? Okay. If you're not clear on what a peer-reviewed article is, you can follow this information here from this Angelo State University has a nice page on how to recognize what a peer-reviewed or referee journal article is to separate it from something like a review article or something like a popular science popular science article that you might find online. Okay, so if you're not clear about that, visit this site and read it over. It's useful to know. All right, so let's get started with today's topics. These are some of the things that we've talked about so far. I like to always start off, or most of the time, kind of getting us back on the same page as sort of like a little mini review. So we said that cancer involves changes at the genotypic level that gradually create changes, greater and greater changes at the phenotypic level. Most of our genome is non-coding and regulatory. Tumor cells, importantly, always start off as normal cells of nearly any type in the body. Just as long as it's a nucleated cell, there is the potential for it to lose control over its genes and therefore lead to disease like cancer. Some of the hallmarks of cancer are loss of differentiation. So the morphology of the cells change, the organization of the cells change, the sheer number of these cells change due to uncontrolled proliferation. Chromosomal defects are kind of at the heart of these changes, either certain point mutations or larger, more gross changes, like entire parts of chromosomes switching around. Can lead to things like amplification of gene expression of certain genes that might control proliferation. Or it can also lead to silencing of other genes that might be responsible for normally inhibiting or preventing cancer from happening, right? If those genes get disrupted, then you lose control. Okay? And we said that understanding the normal cell type of origin of a tumor is key to understanding and elucidating the underlying biology, giving you treatment options. Let's talk about this relationship between stem cells and the many different types of differentiated tissues that make up your body's different parts here. And let's think about what a stem cell is concerned with versus what a fully differentiated cell is concerned with in terms of their goals, their reason for being what they are. So on the left, you have something like a pluripotent cell, right? Sort of the ultimate stem cell that can become any cell in your body. The main goal of a pluripotent cell is to proliferate and replenish all the other types of cells that your body makes. So I said before that in our adult bodies, by the time we're here sitting in cancer class as seniors in college, we don't have any more pluripotent cells in our body, right? The stem cells that we do have already gone down a path of differentiation where they've gotten to a certain point where they can still turn into a handful of different fully differentiated cells, but those are pretty much restricted to what type of multipotent stem cell this is. For example, a multipotent hematopoietic stem cell in your bone marrow can only become the different cells that make up your blood. It cannot become a nerve cell in your brain or a muscle cell in your arm or a bone cell, right? But let's talk about a pluripotent cell that starts off with. It must be tightly controlled in order to very carefully determine, as it's proliferating, what different paths of differentiation the daughter cells that it generates are following. Eventually, through many, many different rounds of cell division and further differentiation, you get to a situation where maybe this pluripotent cell gave rise to this type of cell, this green pentagon here, this type 1, right? We might say it's like this type of cracker, or through a whole different series of differentiation, the pluripotent cell gave rise to type 2, this type of cracker. Right? These fully differentiated cells on the right now, they're not concerned with proliferation anymore. Right? They are concerned with doing the job of whatever type of cell they are, whether it be a nerve cell or a gland cell or a muscle cell. This is a good reminder then that cells typically are concerned with proliferating or doing their assigned tasks, but not both at the same time, because the two processes are incompatible. So consider this. Differentiated cells, some differentiated cells in our body have limited capacity to actually undergo some changes to become a slightly different type of differentiated cell. Some examples are there are certain cells that are supporting your muscle cells, that if you actually have damage to your muscle cells, those cells can actually turn from the original state that they're in to being more like a muscle cell. All right, so we call that transdifferentiation, one type of differentiated cell becoming another type of differentiated cell. Right? But it's relatively rare. Right? There's only a few examples of cells like that in the body. But what if the differentiation, right, actually gets reversed? In other words, if you were to imagine this dotted line, you can imagine undoing the differentiation, the different types of commitments in these cells to potentially bring you back to a state of being a stem cell. Because after all, the genome inside of the stem cell is the same exact genome that's inside of either one of these differentiated cells. All of your genes are there. They have just been switched on or off right, depending upon the differentiation process. So if you undo that, ultimately, in theory at least, you could turn these fully differentiated cells back to a pluripotent cell. But that doesn't happen, right? If you have improper dedifferentiation that happens due to mutations, right, messing up the proper decision process of these of the genomes of these cells, chances are it's not going to be a straight line that takes you neatly back to being a pluripotent cell. It's going to go astray, right? So that's what leads to cancer. And importantly, cancer is not just the disruption of the genes leading to things like dedifferentiation. That's why, remember, we saw one of the telltale signs is the change in morphology, going from well-defined differentiated cells to more generic-looking cells, right? And that's dedifferentiation, right? They're losing some of their differentiated status. Not only is that resulting in a change in morphology, but look, you're going back to this side of the slide where now the cells no longer care about carrying out their normal tasks. They once again get really hung up on this notion of proliferation, right? So this cancer cell then, as these cells have dedifferentiated and gone astray, now this cancer cell, right, because it's preoccupied with proliferation, it then forms a lot of these strangely disruptive cells, which are the cancer cells, right? That even though they are now the result of this cancer cell multiplying out of control, this is not a well-behaved stem cell following careful rules to repopulate all of these differentiated cells. This is an out-of-control cancer cell that's making tons and tons of these weird cells that no longer do their function, but just basically get in the way, right? To the extent that it impacts the health of the tissue, the body, and ultimately leads to death, right? If there's enough of them. So cancer cells have regained the focus on proliferation like a stem cell, but without the tight controls governing how they're doing this. This is the important part about understanding the original cell type of origin that went backwards to get you back to the left side of the screen. Because remember, those cells were the result of many differentiation decisions that were specific, that led you to becoming cell type 1 or cell type 2. Those pathways may still be, to some extent, intact in the cancer cell that represents one of those cells going backwards. So that's why this cancer cell, even though it's out of control, it may still have some dependence on the original cell type specific pathways. And so if you know what those are, you can choose the particular types of therapeutic drugs that have been designed to target those critical pathways and potentially have a much more efficient treatment of this patient's tumor. So we looked at this cartoon before. This is, again, the embryonic cell layers, right? And we said that in the early developing embryo, you have at some point early on the formation of these primordial germ layers, like these tissue layers that then go on to define all the different types of bodies in the tissue. One of these is what we call the mesoderm, right? And the mesoderm will go on to form things like our connective tissues in our body, right? So remember, the connective tissues typically help support the epithelial tissues, right? But the epithelial cells and the underlying connective tissue that supports them are really two different types of cells that depend upon working together in order to make your normal healthy tissues. So tissue that comes from the mesoderm layer, right, in our adult bodies, we tend to call them mesenchymal tissues. So again, these are many of like the connective tissues that underlie your epithelial cells. Why is that important? Because as part of something like an epithelial cell in your body going bad and becoming cancer, remember we said that 80% of solid tumors that we see in patients who present in clinics are of epithelial nature, right? And last time we discussed a couple reasons why epithelial cells are sort of unfortunately set up to be more prone to becoming cancer, right? But an important thing to remember is that epithelial cells, when they're behaving normally, right, they sit on this basement membrane that separates them from the connective tissue beneath. Right? And we looked at this image last time of malignant cancer forming, where the epithelial cells are breaching or breaking through that basement membrane and getting into the connective tissue beneath. So in many types of tissues where you have the boundary between connective tissue and epithelium separated by the basement membrane, that connective tissue beneath we call the stroma. Right? So different terms here, right? Stroma is typically connective tissue underneath epithelial tissue, helps support the epithelial cells normally, right? Not only physical structural support, but also sending all kinds of messages and things to the epithelial cells to tell them how to behave. Right? And that stroma, that connective tissue, is an example of mesenchymal tissue, right, that comes from the mesoderm layer of the embryo. So why is this important? Because as the epithelial cells start going bad, right, they have to learn how to no longer behave as epithelial cells. Right? And part of them being able to penetrate and spread has to do with them undergoing mutational changes that make them less epithelial and more mesenchymal so that they can actually survive and function in a connective tissue like stromal environment. So cancer cells undergo this epithelial to mesenchymal transition, or EMT. Right? Later in the semester, when we really study the process of malignancy and the spread, right, of tumor cells from the original primary tumor site to a metastatic site. So when we study the process of metastases, we will look at EMT in more depth. But I think it's good that we have an introduction to that to start off with. Right? So malignant epithelial cells may take on the properties of underlying tissue. Right? Part of that is changes in morphology that allow them to actually function in more of a mesenchymal tissue environment. So changes in their plasticity, right, their morphology. And these are progressive changes that pathologists use when they look at a biopsy slide from a tumor. They use these different visual cues about the changes in the plasticity or shape of these cells as part of the prognosis of this patient, right? For example, terms like benign versus malignant, but in cancer oncology terms, I think you've heard about staging, right? The different stages of cancer progression from stage 1 to stage 4. Right? A lot of that has to do with what the pathologist sees in the plasticity. So let's talk about this. Different terms having to do with plasticity. Hyperplasia is an early hallmark of tumor growth. So you can tell just by the name. Hyper means like, right, a lot of something. So one of the first signs is simply a dramatic change in the number of these cells, even if the cells themselves at this point still look kind of normal, right? Suddenly you have out of control proliferation, and you see a lot of them. So this is termed hyperplasia. Let's look at that here. So here is a normal cross-section of mammary gland. So most of the whitish space in here is the stromal or mesenchymal tissue. And
Dr.
what you see, yeah.
The slides froze.
Oh, okay. Thank you. Let me stop the share for a moment here and start it up again and see if it works better. Okay, can you see it now? It says hyperplasia is an early hallmark of tumor growth.
Yeah, I can see it now. Thank
okay,
you.
Good, thanks. All right. So thank you for that. So here you see cross sections of glands, right? These are the mammary glands that actually consist of glandular cells that are making milk, right? And these are the cross sections. So these glands, if you look zoomed in, these are the glandular epithelial cells that line the inside of this duct, right? So it's a gland, so it has a duct that helps collect the milk and transport it away. And the lumen, right? So the space inside of this duct where the milk is made and then flows away is the lumen. right? Just like our intestines, right, have a lumen in which is the space that all of our food flows. So if this is the normal sort of appearance of it, if you look at this over here, you can see this is a hyperplastic mammary gland duct. Right? The reason is the individual cells are still pretty well defined. They still look like they're supposed to look, but there's just way too many of them. Right? So this is classic hyperplasia. Right? Is this cancer? No. Right? Is this the start of tumor formation? Yes. Okay. But at this point, it is not showing the signs of being true cancer. All right. Dysplasia, then, is when you actually start seeing changes in the cell shape. So in this case here, this is another nice example of a tissue cross-section where they've able to capture different regions showing the progression of disease. So on the left side, I'm not sure, I think this is maybe esophageal or some kind of GI tract tissue. But you see here, these are the epithelial cells, okay? The dotted line represents where the basement membrane would be that they sort of depend on to separate them from the stroma or the mesenchymal connective tissue beneath. As you progress to the right side, not only do you see clear evidence of hyperplasia, right? I mean, it's clear as day, right, that there's tons more of these cells, but you also begin to see that some of these cells are changing their shape. Right? So rather than having, like over here, these well-defined kind of cells, these are now beginning to get kind of strange, right? Especially these ones I think that the arrows are pointing at. They look very different than the normal healthy ones. So we say that things are getting a bit dysplastic now, right? Metaplasia is another interesting phenomenon where you actually start seeing tissues appearing in the wrong place. So this is definitely an example of the esophageal lining. The esophagus, from your mouth to the entrance to your stomach, the epithelial lining of it is designed to be, pretty resistant to the physical stresses of food that you're eating, right? I mean, as you're eating food, right? Yeah, you chew stuff up, but when you swallow it, it can still be pretty rough, and it can actually scrape against the sides of your esophagus, right? So your esophageal lining has a lot of mucosal glands that produce a lot of mucus to make the food go down easier. Once you enter the stomach, the lining changes because the stomach, of course, has a different function, right? It's going to be concerned with generating a lot of hydrochloric acid and things like that. Where the junction happens between the esophagus and the stomach, right? It's a pretty quick transition in the nature of the epithelial lining. So what can happen then is as changes start taking place in, let's say you have here the beginning of disease in the stomach lining, all right? In this case, some glands of the stomach are beginning or have turned into cancer. So again, right, they are carcinomas, right? And because they're glandular in origin, they are adenocarcinomas. Right? In this case, they're ulcerated, meaning that they've actually penetrated the barrier of the GI tract, right? And you have a lot of inflammation and stuff like that. If you look at the very left here, this is what the normal epithelium of the esophagus looks like, right? So you can see here, right, this is sort of your protective layer with a lot of mucus above it. I think all these little specks, all these little grayish specks appear as actually mucus. Right? You can see as you get close to where the stomach is, you begin to notice that you have examples of stomach lining actually part of the esophagus. Right? So in this case, metaplasia is kind of like the tissue is beginning to improperly occupy different regions that they shouldn't. So this is an example of what we call metaplastic Barrett's epithelium, right? Where biopsies from the epithelial lining already show evidence that they're becoming more like the lining of the stomach. Okay? Anaplastic is like an extreme. All right, so we said that dysplastic just means things start looking funny, right? Anaplastic means things are so wrong now in terms of the shape of these cells that when a pathologist in the clinical lab looks at this under the microscope, he or she can no longer tell just by sight, what they're looking at, right? What kinds of cells these are, what kinds of cells did these start off as, right? And so that's the big challenge. And obviously, if you get to this point, then the prognosis wouldn't be as good as if you were just looking at a slide and it looked like you still had well-defined cells that were just maybe a bit hyperplastic, right? Anaplastic means, this is clearly now progressed, and things can look pretty serious. So here is a summary then of these terms. So one term that I didn't mention explicitly, but I will now, is neoplasia. Right? So any change in your tissue's normal phenotype can be thought of as something new, right? So new changes in tissue growth, we call that neoplasia, right? And so neoplasia is kind of this umbrella term because hyperplasia, dysplasia, anaplasia, right, are all part of this big neoplastic change in the tissue. And then metaplasia is improper cell types replacing nearby tissues. Okay. As these cells turn into tumors, right, and as these tumors start proliferating and becoming bigger, a question that in the past, I think when cancer researchers first started studying tumors, one of the big questions was, do tumors actually represent one group of cells that keep proliferating and becoming a big mass? Or are they actually a group of different cells that all started proliferating out of control and then fused together to form a big tumor? Because a big tumor, a solid tumor, oftentimes is very heterogeneous. What do I mean by that? It's not going to be a uniform piece of tissue that, depending upon where you take a biopsy from this tumor, it's going to look the same. I think you guys can already figure that out, right? Big tumors are messy, disgusting, heterogeneous things, a big mess of different cell populations. So you can see how early on it wasn't clear. Is that the result of different types of cells starting off, all going bad and then fusing together? Or is it actually one cell that started going bad and in the process of controlling out of control, different pockets or subpopulations of those cells each started going off in their different direction to create a heterogeneous mass? It turns out tumors start as monoclonal growths. Right? An individual cell that goes bad or rogue, I like to think of it that way, right, eventually leads to a tumor mass that may lead to becoming cancer. So how did researchers finally determine this? So early on, they did a pretty elegant, I think, experiment, right? And they said, in an early embryo, right, so in females, right, you have two copies of the X chromosome, one from mom, one from dad. And I think all of you know that there is this phenomenon known as X inactivation. That is, as the stem cells begin to proliferate and become different types of differentiated cells, or generating the different types of differentiated cells in your body, right? They need to switch off one of those X chromosomes so that you have the correct complement or the correct number of those genes that are on that X chromosome, right? That process is more or less random. So in an early embryo, let's say one that's only made of something like, a dozen cells, both mom and dad, so maternal and paternal X chromosomes, are active, right? So that's why they're capital M, capital P. But as these cells begin to develop into the embryo, right, these individual cells begin to randomly switch off either the M, making it lowercase, or they randomly switch off the P, making it lowercase, right? So you would start having this sort of combined population of which X chromosome is actually the one being used. As a result, in our adult tissues, right, females have a mosaic of different tissues that are using the X chromosome from dad and different tissues that are using the X chromosome from mom. I think all of you know this, but I always think that's amazing when you think about it, right? That means your body, even though you don't see it visually, right, is actually made of different sort of patches of cells that are using dad's X chromosome versus mom's, right? Of course, in men, that's not relevant at all. We only have the X chromosome from mom because dad gave us a Y chromosome, right, which is the source of all of our problems. But anyhow, so in an adult body, then, if a tumor starts forming, what they found was that the resulting tumor that formed, even though it might be really ugly and messy and heterogeneous with all kinds of weird shaped cells, all of those cells would have the same status in terms of X inactivation, right? Either the lowercase m with the capital P or the lowercase p with the capital M. So that is compelling evidence that all of the cells in a big tumor mass probably came from one starting cell that was going rogue. Okay. Another area of this class that we will talk about is understanding how different types of normal cellular processes inside the cell, right, are disrupted by things like mutations, and how randomly coming up with mutations is very much part of the natural selective process of a cancer cell gaining additional mutations that it carries with it as it becomes more and more aggressive. Right? So these random mutations then, because they are random, right, could either disrupt certain types of normal functions in the cell if that gene that the mutation happens in, right, let's say is vital for a particular pathway. If you lose that gene, that could actually hurt the ability of this cancer cell from continuing on, right? Or certain mutations by chance might enhance something within the cell that gives it an advantage to continue on. Right? Here's one example, just to kind of introduce this to you. Glucose metabolism in the cell. In a normal cell, you guys all know this, on the outside of the cell, the extracellular space, glucose is received by certain types of glucose channels in the membrane, like the GLUT1 transporter that allows glucose to come in, right? And you remember from your bio 275 days, glycolysis happens in the cytoplasm, and glycolysis is how you get a couple wimpy ATPs out of this, and you end up with pyruvate, right? And pyruvate then goes into the mitochondria, where it's converted into acetyl-CoA that then feeds into the Krebs or citric acid cycle, which gives you buckets of ATPs, right? When you finally use all of the electrons that are generated by the citric acid cycle to drive ATP synthesis, right? So a normal cell heavily depends upon the citric acid cycle to get most of its ATPs. What happens then? So sorry. So you guys all know, right? This only works because you have oxygen present. This is aerobic respiration, right? It's oxygen that allows the electron transport chain to actually make these ATPs. So what happens then if you have a large tumor mass that's getting bigger and bigger and bigger, and the blood vessels that supply things like oxygen to these cells are getting progressively pushed farther and farther away from the cells in the middle of this tumor? That means the oxygen available for the cells in the middle of this tumor, right, gets depleted. And if those cells can't find a way to cope with that, they're going to die. And in fact, that's true. In most large tumors, one of the things that makes them really disgusting and gross is they're filled with necrotic cells, cells that could not continue living because they were cut off from oxygen. Right? There are, though, absolutely examples of tumors that really begin to grow big and out of control because the cells have stumbled upon different mutations that get around this dependence on the citric acid cycle and oxygen. For example, all right, certain mutations in different proteins that support or drive glycolysis if those mutations happen, so these are just some examples of genes that we will study this semester. So I like this because it introduces you to these, but don't get scared because you're like, what are these, right? We will study these, right? But these are examples of genes that oftentimes are mutated in the progression to cancer. And these are genes that are involved in things like, for example, the expression of GLUT1. So if all of a sudden the cell is making way more GLUT1 transporter than it needs, it's actually taking in a lot more glucose than it needs, right? And that would drive glycolysis higher. Or some of these genes, the protein products of these genes, are directly involved in the proteins that are part of glycolysis. So really amplifying or ramping up the glycolysis. It doesn't mean that all of a sudden one glucose molecule makes more than just 2 ATPs, right? But what it does mean is that you have a ton more glucose coming in and a ton more of the proteins that are ready to make glycolysis work. So you actually get a lot more ATPs just from glycolysis. And these cancer cells no longer need oxygen because they've shifted from aerobic respiration to anaerobic respiration. And then if they stumble upon the right combination of these mutations, right, suddenly now they can continue growing and the tumor will then become very aggressive. Early on, the researcher, his name was Warburg, who identified this phenomenon, thought that somehow the cancer cells were now doing glycolysis in an aerobic way, right? So he termed it aerobic glycolysis, but that's very misleading, right? Because the whole point is that these cancer cells have mutated so they don't need to depend upon aerobic respiration, okay? So this is just one example of many that allow you to begin to think about how mutations that happen in the right combinations, if they involve the right set of genes, can end up changing the physiology of a cell to give it an advantage, a growth advantage. So where do these mutations come from, right? And how is it that scientists even made the connection that it's all about mutations? I think all of you know that mutations are the basis of cancer, but then that's kind of funny because that's you guys as college students in 2021, right? Think about college students in the 1800s, because hey, look, right, ever since the beginning of the human race, there's been cancer, right? So medical researchers, biomedical scientists, they were aware that cancer is this problem, but they didn't know what the source of cancer was. So even though for you, yeah, you easily understand, yeah, it's mutations in genes, they didn't know that, right? So early on, a researcher named Ames tried to identify what were the things that actually influenced the creation of cancer. And he did this very famous experiment known as the Ames test. He took many different substances that were believed to cause cancer, right? And he treated, I think in this case, he was using bacteria. So he would treat salmonella bacteria with these different types of toxic, compounds and things. And he would ask, right, what would happen to these different bacteria in terms of their ability to grow under conditions that normally the bacteria wouldn't? And what he found was that he could order or put into sort of a ranking the compounds that that the bacteria seemed to respond very easily to, and something changed in those bacteria to make them now easier to grow and grow more rapidly, compared to compounds on this end where it took a lot longer or higher doses of that compounds in order to make those changes happen. Okay. And what he did was he measured that on the y-axis by saying that this somehow has to do with the carcinogenicity, right? The ability to actually stimulate cancer in things like, mammals, right? So one of the most potent carcinogens that Ames determined was this aflatoxin, B1, right? Because it took a very little amount, just a few 10 to the minus 2 micrograms in order to actually cause mutations in the salmonella, right? And it turned out that people knew that aflatoxin in things like animals was also very, very carcinogenic. Right? So this was a particular interest to him because he saw that what was happening inside the salmonella was that there were mutations happening. So let's look at this aflatoxin. So it's actually a fungal toxin. I should say, right, that the incidence of cancer across different populations of people is certainly not even. You might think, hey, we're all human beings, right? We have a human genome, and therefore we probably respond the same to different types of carcinogens or mutagens, whatever. But that's not necessarily true. This table here really highlights the different cancer frequencies across different types of populations. For example, here are different types of cancer sites like skin, prostate, stomach, ovary. And these are countries that show populations at the highest risk of developing these, contrast to countries of lowest risk. Right? So for example, something like skin cancer, people in Australia, right, they are at the highest risk of developing skin cancer, whereas in places like Japan, we see the lowest incidence of skin cancer. But on the other hand, this doesn't mean that people who are in Japan are protected against cancer somehow, right? Because you can see Japan actually has some of the highest incidence of stomach cancer. So why is that? Here is some data. It's from your textbook, but I think this is really nice data because it actually relates to the Hawaiian Japanese immigration story. Right? If you were to look at a group of people who were Japanese nationals who lived in Japan from 1970 to 1971, right? Or from 1988 to 1992, and you looked at a population of people who immigrated to Hawaii during those times, 1988 to 1992, or older, 1968 to 1972, that's what these colors are. And what you're looking at is the rate of stomach cancer in these individuals, as well as other types of cancer like prostate, colon, and breast, right, compared across these different populations. And you can see that something like prostate cancer, people who are Japanese nationals living in Osaka, right, had much lower incidence of prostate cancer compared to people who immigrated to Hawaii, right? So the green and the blue, right? And in comparison, the orange are Hawaiian Caucasians. So that gives you sort of a reference point here to what is actually considered high versus low. So for colon and breast, it's a similar type of pattern, right? The Japanese living in Japan had lower rates of these types of cancers than those who immigrated to Hawaii. Right? The one that's very striking is stomach cancer. Okay. In stomach cancer, the Japanese living in Japan by far had much, much higher rates of stomach cancer compared to the immigrants who came to Hawaii. And look at the rates in Hawaiian Caucasians are the lowest. So the question is, what is going on here? What changed? And it turns out it has to do with these aflatoxins. So back in Japan during the early 70s, right, they were still storing things like grains, like rice, in large sort of old-fashioned silos. And these silos oftentimes, because they were old, did not have good ways of keeping moisture out. So as a result, you had a lot of fungus that would grow on these rice. And these fungus would produce these aflatoxins. So for immigrants who came to Hawaii during this time, right, the thought is that certainly these days, right, we have much better ways of storing rice to sort of discourage the growth of these fungus that form these types of toxins that cause cancer. So here you have an example where genetically speaking, there's not much of a change between the types of people you're talking about, whether they live in Japan or they immigrated to the US. But what we're talking about here are changes in exposures, right, that can be brought on by just being in a different geographical location. Right? So that really is, I'm going to end there talking about that today, because exposures then is another piece of the puzzle when we talk about cancer. Not just the changes that are taking place inside of the genome of a cell as it goes bad, but also the influence of things from the outside potentially driving the growth of cancer in combination. Okay, so let me go ahead and stop there today. Let me turn off the